The next topic is gas exchange in the lungs. So here we see the bronchiole with the terminal bronchioles ending in the respiratory bronchioles with the alveoli, right? And each of these little balls represents an alveolus. And what I'd like to draw your attention to now is the arteries and veins that are interconnecting and intertwined between the tissues of the lung, and most importantly, how they wrap around the alveoli. And if you look over here, here's one alveolus, and I want to now show you a close-up of the blood capillary network overlying one alveolus. Here we see the capillaries, right? On the top, this is going to be capillaries having oxygenated blood. And then down here, we're seeing capillaries having the deoxygenated blood. So usually in most tissues of the body, we think of the arteries as having the oxygenated blood and veins as having the deoxygenated blood. The exception is in the pulmonary arteries and pulmonary veins. So what we actually have in the pulmonary arteriole, which is what we're seeing here, that goes down to the pulmonary capillaries, this is the arterial capillaries in the lung. They are carrying deoxygenated blood from the heart, right? That's pumped from the right ventricle into the right pulmonary artery, and that will then enter the lung. So basically, the arterial blood in the lung is going to be deoxygenated, and as they're in the lung, they will leave being oxygenated. So this is actually pulmonary venules that are oxygenated, right? And that's going to take the pulmonary venule is going to go back to the pulmonary vein to bring the oxygenated blood into the left atrium of the heart, so then it can be sent to the left ventricle for systemic circulation. So I want to show you that there's a huge surface area to facilitate the gas exchange that is so critical for mammalian life to continue. What I'd like to show you now is the concentration of gases in blood as they circulate the body. So let's start from the pulmonary artery into the lungs. So the blood coming from the pulmonary artery has a pressure for oxygen of 40 millimeter mercury, whereas the pressure for the carbon dioxide is going to be 46 millimeters mercury. That's in the blood in the pulmonary artery that is deoxygenated. Now, if we look in the alveoli, we'll see the oxygen concentration and carbon dioxide concentration reflect the concentrations of these gases in the air. And for oxygen, we see the pressure is 105, for carbon dioxide is 40. So we've learned in this class and by other classes that molecules tend to go from a high pressure area for that molecule to a lower pressure. So since 105 for oxygen is more than the 40 in the blood vessels in the pulmonary capillaries, in the arterial pulmonary capillaries, there's going to be a net movement of oxygen from the alveoli to the capillaries. Because we see the pressure for the carbon dioxide is 46 in the capillaries, it is 40 in the alveoli, there's going to be a net movement of carbon dioxide from the capillaries to the alveoli. And this movement is shown over here with these arrows. Then, leaving the lungs, we have the oxygenated blood with less carbon dioxide in it that is going to be entering the pulmonary vein. And we see that the pressure for oxygen went up to 100 millimeters mercury, and for carbon dioxide, it actually went down to 40. This blood will be going to the left atrium and left ventricles, oxygenated blood to be pumped out to the aorta, and then the arteries, and then sent to capillaries. In capillaries, there will be a net movement of oxygen from the capillaries into the body cells that need oxygen as a final electron acceptor, and we'll be seeing a net movement of carbon dioxide, which is produced as a byproduct of metabolism, and that'll be picked up by the blood in the capillaries. And then we see that there's a huge change in the pressure of oxygen and carbon dioxide. So the oxygen level, we said, was about 100 millimeter mercury. It's going to go down to 40, and this is under resting conditions, just normal conditions, not exercising or anything. And then we see that the carbon dioxide concentration is going to go up from 40 to 46. And again, in both the lungs and in the tissues of the body, the gases are just diffusing by pressure. So that gases will just go from a higher pressure to a lower pressure, and from basically a higher concentration to a lower concentration, right? It's going from high pressure to lower pressure. And remember that this is really critical for diffusion. And diffusion occurs, right? Diffusion always goes from a higher concentration to a lower concentration. So this is just simple diffusion, and we're seeing that happen in both the lungs and in the tissues. And this exchange is always going to happen in capillaries because they lack the tunica, the three tunica layers that would prevent the rapid diffusion of these gases. So the next thing to think about are hemoglobin and oxygen transport. So the plasma oxygen concentration in the systemic arteries is about 0.3 mil per 100 ml of blood. And the total oxygen content of blood depends on the pressure of oxygen and the hemoglobin concentration. So what I'd like to show you now is the oxygen content in plasma and whole blood. So if we look at plasma, and of course this is an experimental system using a gas tank, creating an artificial situation, but it basically mirrors what is happening in the body. So what we have on the left is plasma having an oxygen content of 0.3 mils of oxygen per 100 mil. And I want to notably point out that we have 1, 2, 3, 4, 5, 6 molecules of oxygen, shown in blue, that is yielding this concentration. So when we talk about the pressure of oxygen, it's based on the number of free molecules floating in a system. If we look in whole blood, we actually see that the concentration of oxygen is 20 mils oxygen per 100 mil of blood. And most of the oxygen is actually bound to hemoglobin, which is hemoglobin shown in red. And each hemoglobin has the ability to bind 4 oxygen molecules. And I also want to point out that if we look at the number of free oxygen molecules, those little blue dots, we see there's 1, 2, 3, 4, 5, and there should be a 6th one somewhere. And then there, because basically, if you take out, the reason that we have the pressure of oxygen being 100 millimeter mercury in both of these compartments is that the pressure of oxygen only looks at the free oxygen floating in the solution. So in blood, the oxyhemoglobin is actually located inside of red blood cells. So that the oxygen is not detected when we talk about the pressure of oxygen. The pressure of oxygen we're just talking about is the free oxygen in the blood and not the one in the hemoglobin. So the point is that the majority of oxygen in blood is actually carried by hemoglobin, and the very small minority is actually found in plasma, which is free-floating oxygen molecules. The majority, again, is put in is attached to hemoglobin. And the interesting thing is, if you were to take all the red blood cells out of here, and you could measure the pressure of oxygen in both of these compartments, if you took all the red blood cells out, it would still be giving you the pressure of 100 millimeters mercury. Because when we measure the oxygen pressure, it only takes into account the freely floating oxygen that is dissolved in water, which is the plasma, and it doesn't measure the amount of oxygen attached to hemoglobin that is found inside of red blood cells. So what I really want to highlight here is when we talk about the pressure of oxygen, that's really looking at the oxygen dissolved in plasma or the water or blood, and it doesn't measure the oxygen that is carried by hemoglobin. So red blood cells, as you know, are called erythrocytes. They are flattened biconcave discs among the smallest cells in the body. They critically lack nuclei and mitochondria. We have so many of them. We have 5 million per cubic millimeter of blood. They live for only about 120 days. And listen to this. Each red blood cell contains about 280 million hemoglobin molecules. That's right, 280 million hemoglobin molecules in one red blood cell. This gives red blood cells an amazing ability to be carrying oxygen. And when a red blood cell is done with its 120-day lifespan, the iron heme needs to be recycled from the liver and spleen, and it will be carried by transferrin in the blood to be going back to the bone marrow because it's very energetically expensive for the body to produce this. So our bodies recycle the heme, iron heme, so we don't have to keep recreating it. This is a diagram of hemoglobin, and hemoglobin is composed of 4 polypeptide globins, 2 alpha and 2 beta. Here we see 2 alpha, there's 2 beta chains, and I want to reiterate again that each hemoglobin can carry 4 molecules of oxygen, and there are critically 280 million hemoglobin in the average red blood cell. This allows each red blood cell to carry over 1 billion oxygen molecules. And then there's the iron in the heme that's critical in hemoglobin. So hemoglobin, as we know, can bind oxygen. We call that oxyhemoglobin. And the reduced form of iron, Fe2, can bind easily to hemoglobin. We also have carbaminohemoglobin, and that's when hemoglobin is bound to carbon dioxide. So this is great. So hemoglobin can carry oxygen from the lungs, and it can also help carry carbon dioxide back to the lungs. And we can actually detect if hemoglobin is binding oxygen or carbon dioxide by using a pulse ox, which measures the absorption spectrum of the different kinds of hemoglobin.